Regulatory compliance is not about following rules. It's about guaranteeing quality in every dose delivered to patients.
There are several checks and balances in place in order to ensure that the products we manufacture are safe for patients.
Innovation in compliance practices is critical. Going beyond compliance is important. There are many ways that you can do this.
PUPSIT is a requirement for European regulators, and I will explain you how this requirement can be met.
I'll tell you about the regulations that govern the fill-finish process.
If you follow these guidelines, you'll do well.
So in the United States, sterile manufacturing is covered by the 21 CFR, which is the Code of Federal Regulations, Part 210 and 211. CFRs are general frameworks of guidance from the federal government. 210 and 211, as well as the draft guidance for aseptic processing, is really mainly when we're making drugs through our aseptic process, is what we adhere ourselves to. Specifically for Berkshire Sterile Manufacturing, we're governed by those and we're audited against those for FDA. For 21 CFR Part 210 and 211, deal mainly with how to manufacture and distribute safe drugs and therapeutics, how facilities are designed, how utilities are used, training, validation, testing, things like that. It's a bunch of frameworks in order for the drug manufacturers to produce drugs in a safe and effective manner. In Europe, they go by what's called a EudraLex, and the EudraLex Volume 4 is what really governs the medicinal oversight of the European Union. Specifically for sterile manufacturing, Annex 1 is the benchmark by which manufacturers and distributors are measured against for regulations. The Europeans also have something called a qualified person, or a QP. In the United States, someone like me would review the final batch record, certify that testing was completed, met specifications, and can be released for distribution and use in clinical trials, or if it's commercial, released for commercial use. In Europe, a qualified person would be that individual who is certified and under the regulation of the European country they're in. So if someone in France, they would have to be registered with the French government, someone in Germany, et cetera, et cetera. And they would be the ones that would be certifying the batch. If we made it to go to European distribution, they would be looking over all of that too, and then certifying the batch for release into the EU market. Mostly what's covered in the 210 and 211 regulations are covered in Annex, although there are some differences. Annex 1 is more of a focus on pharmaceutical quality management, where they like to see your quality risk management baked into your entire process, right? Looking from start to finish. And the pharmaceutical quality management system that Annex 1 is wanting those individuals to undertake really requires you to have a robust risk management system, a good CAPA system, really trying to be proactive in looking at risk instead of being more reactive when things may happen. In addition, there are some differences in relation to operations. PUPSIT is a big one. So PUPSIT is pre-use post-sterilization integrity testing.
This is the requirement by European regulators based on the recent Annex 1 revision which happened last year. We used to do before a pre-use testing and post-use testing, but based on these recent guidance, we have to do a testing between a pre-use and the post-use. When we're sterilizing the filters, before it gets used in the formulation process, we need to perform the integrity testing on those filters to make sure that the sterilization process has not impacted the integrity of the filter. The major concern from the regulators are that sterilization of the filtration can cause microscopic defects into the filter. And what happens with that is, like, when we do the filtration, the formulation can mask those defects. And when we do the post-use testing, we can get a false positive result on the filter integrity. And this is the reason we need to apply PUPSIT so that we don't have a false positive result, and ensure that before the filter is being used, after the sterilization has been performed on the filter, there are no defects on the filter. PUPSIT is performed by using specialized assemblies, which we need to design. We use barrier filters. So those barrier filters are filters which are composed of like both hydrophobic and hydrophilic layers in it. So that is to allow the air and the Wi-Fi, which is used for the integrity testing, to go out of the line. And then along with that, it also uses an hydrophilic filter that connects to the line, which goes to the integrity tester. So this is the setup of the assembly, and the testing on the PUPSIT assembly is performed in the same way using the recommended filter integrity testing parameters. Those are given by filter vendors. The most important thing to understand with PUPSIT, when you go to the post-use testing, you not only test the filters which are used for the filtration of your formulation, you also need to test all these barrier filters which are present on the assembly. It was in like 1997 when European regulators first proposed to apply PUPSIT, but again it was a recommendation. And now in the recent Annex 1 revision, they have used the term "should" into the statement. Regulators are aware of the concern that PUPSIT cannot be applied in all the processes. If you cannot apply PUPSIT on all the processes, you have to do a risk assessment and provide a justification why PUPSIT is now required. PUPSIT is prone to dilute the formulation. So if you're handling a very low volume formulation, then that could be justification of not applying the PUPSIT into your manufacturing process.
The other thing in Annex 1 that BSM is ahead of the curve on is that Annex 1 is pushing more and more manufacturers to more advanced technologies, isolators, RABs, which are restricted access barrier systems, right? Trying to take as much of the human element out of the manipulations as possible to ensure a greater sterility assurance. Isolator technology really is the gold standard. The reason for that is, you have a more controlled environment. You're taking a lot of the human manipulation out of it. You still do have some human interactions, but they're restricted by a barrier through the isolator using gloves. You're really not touching the product. Whereas if you're in an open manufacturing environment, the entire area along with the operators are in play. And individuals have to be plated. You have to be, extremely careful with your aseptic technique. Moving forward to next generation with robotics and automation, coming into a robotic line with Steriline, that further removes the human element from the processing, which in turn increases the sterility control that you have for a product. So really the entire industry is moving towards isolator technology, RABs, restricted access barrier systems, to eliminate the human element and to increase the quality of the product. And one of the other things that Annex 1 focuses on is what's called a CCS, Contamination Control Strategy, which any reputable biotech pharmaceutical company will have. It may not be in one document, it may be in different portions, but it's the contamination control and the risk mitigation of any potential contaminants that may occur. As far as, different plating.
We're required to demonstrate a grade A environment within the isolator. That's done through particulate monitoring, viable air monitoring, and viable surface monitoring. Non-viable particulate monitoring occurs throughout the fill, meets a grade A requirement for particulate. Viable air monitoring also occurs continuously through the fill through samplers in each chamber.
US doesn't require passive monitoring. Annex 1 and EU does during manufacture.
For European Union customers, we'll perform passive monitoring within the fill chamber. And then at the end of the fill, we'll do surface monitoring. Surface monitoring will be contact plates throughout the whole chamber. It'll cover all the major surfaces, including glove, fingertips, and wrists. And then critical surfaces that include transitions between chambers, sampling on the fill parts, like fill needle and stopper ball. It's a separate plate for every sample site. So we'll see data for each location, including the fingertips of each glove, the wrist of each glove, and major locations within inside the isolator.
We also do active monitoring during formulation for an EU product. US is not required. And then as far as release testing and process testing, there's the USP test versus the EU pharmacopoeia. But through harmonization, global harmonization, a lot of the testing that we do, regardless of whether it's EU or US, USP, the testing doesn't require anything different due to that harmonization.
The requirement for a quality control unit is defined in the Code of Federal Regulations. A quality unit consists of the responsibility to inspect, test, and disposition materials. BSM breaks it out into testing and inspection responsibilities with the quality control unit, while QA is responsible for disposition. The quality control team is responsible for developing as well as testing materials. These range from incoming materials at BSM, or in-process samples during drug product manufacturing, of course, finished product and stability. Material release testing refers to testing we perform on incoming materials or starting materials for drug products. Typically, we test against a manufacturer certificate of analysis or compendial regulations, such as a USP NF monograph or a European Pharmacopoeia monograph. The release testing is typically for the identity, strength, purity, and quality of the drug product. Of course, these can be accomplished in many different ways.
So our minimal requirement is going to be sterility testing. At a minimum, we have to demonstrate sterility. My group also will perform endotoxin testing, so we're looking for pyrogens.
Quality can be addressed with standard compendial tests such as subvisible particulate, endotoxin, and sterility. The reason for doing the release testing is to ensure we manufactured what we intended to, as well as ensuring that the product is safe for the patients who will be using it. This is also a requirement in the Code of Federal Regulations under good manufacturing practices. Release testing doesn't necessarily have to be performed by the CDMO manufacturing the product. With the complexity of modern medicines, it's not uncommon for some laboratories to have expertise in some type of methods. Because of this, sponsors of drugs and owners of drugs can often use several different laboratories to perform all the necessary release testing. Material release requirements do differ depending upon the phase. For phase 1, material requirements are less strict. We need a C of A indicating the material is released and has met all criteria, and BSM is obligated to verify the identity with at least one ID test. For later phase, beyond that, that is where BSM not only needs to ensure the identity of the material, but its strength, purity, and quality as well. Verification applies to a situation in where you're showing that a material, a sample, a product can be tested with an established method. This is typical for compendial testing, where the USP or European Pharmacopoeia has defined testing, and you're just showing that your product can be tested and is suitable in this type of method. Regarding qualification and validation, we often use what we call a phase-appropriate validation. So for earlier phase, prior to phase 3, we'll follow the guidance for the ICH has for method validation. However, we may not do every characteristic. For example, robustness may not be examined during an early phase qualification. However, it would be included in the standard validation package.
So CCIT is, it stands for Container Closure Integrity Testing, which is a type of test designed to determine if a container is sealed properly. Within the United States, one of the main ones is USP 1207, which is a general chapter that explains what CCIT is and then gives some background information on the different methods that are out there, how to determine which method to use, as well as how to properly validate the method. There's also USP Chapter 382, which is not yet effective. It becomes effective December 1st of 2025. And that is on the functional testing of elastomeric container closure components. This chapter makes CCIT a requirement to perform package validation studies for any sort of sterile packaging system that contains basically a rubber component, something like a stopper or a plunger. Annex 1 does have information about CCIT, and it requires CCIT as well, but the language is pushing towards the use of deterministic methods as opposed to historically used probabilistic methods. There are a lot of different methods to perform CCIT. Old kind of historical methods that are probabilistic, such as dye ingress, and the industry as a whole is trying to move more towards the use of the deterministic methods, some of the newer technologies such as vacuum decay, helium leak, laser headspace, and HVLD. They all have their pros and cons, things that they're really good at, things where they have limitations. And so selection of which method you're using is really dependent upon what your container closure type is, as well as what your product is. And then also your intended use. Are you doing a package validation where you need to verify your container closure system can meet the maximum allowable leakage limit? Or are you more interested in a routine test for a stability study and not needing to prove that maximum allowable leakage limit? Well, CCIT is a more holistic test than sterility. Sterility tells you if bacteria is able to grow in the product itself. Whereas there are other things beyond bacteria that can impact product. There are products out there that are sensitive to oxygen or carbon dioxide, and if those gases get into the container, they will affect the product quality. If you have a leak that allows liquid to come out of the container, you could have failures for things like fill volume, and a sterility test won't determine that either. Sterility testing also has a higher likelihood of false positive and false failures. So the new techniques, the new methods that are out there for CCIT, these deterministic methods have a lot lower false failure, false positive rate. Many of the methods are also non-destructive, unlike sterility testing. So if there is a failure, the investigation can be performed and actually those same units retested to verify whether or not it was a true failure. Best practice would be absolutely to implement CCIT as part of a stability program. The FDA published a document back in 2008 specifically suggesting that CCIT be used in lieu of sterility testing as part of a stability program. The industry is moving in the direction of a holistic approach to container closure system.
Some of the practices that should be avoided when working within an aseptic area, whether it be an isolator or in just an aseptic environment as a whole, rapid movements, moving hands and materials over open containers, not following proper cleaning practices, wiping down gloves, wiping down implements that are already sterile to ensure that the sanitization remains, not putting down certain implements on surfaces within the isolator that could pick up potential particulate or contaminants that might not have been picked up during the VHP process, things like that. Just being aware of your movements and how those movements may impact the product from a contamination standpoint and also a particulate standpoint. On the floor QA, or shop floor as we call it here, it's a partnership that quality assurance has with manufacturing. The individuals in that team work hand in hand, side by side with the manufacturing operators during GMP operations and filling. They're in the rooms during formulation, they're in the rooms during filling, and we're really there to ensure that the operators in manufacturing are performing their jobs at the highest quality level. We're providing quality oversight and compliance guidance during the times of filling and formulation. When issues do arise, they can be handled on the floor. Conversations can be had, deviations can be opened in real time, comments can be made, escalations can be done. We can control issues in the moment rather than letting them sit around and having to deal with them later. Quality should always be top of mind.
We need to constantly seek ways to push for excellence in compliance in all aspects of manufacturing.
These regulations which are listed in Annex 1 are not the hurdles, but these are kind of milestones.
When we prepare methods, our goal is to ensure that they stand the test of time and stay ahead of regulatory requirements.
We have a responsibility to patients to ensure that every drug manufactured at Berkshire Sterile is safe and effective.